All right, this time we're talking about the heart, the pump for the cardiovascular system. So starting with the heart itself, there's four chambers, of course, composed of cardiac muscle tissue, the only muscle tissue of the heart, the only location of cardiac muscle. And with the four chambers, it's a dual setup. So there's the right and left superior chambers, known as the right and left atria. And then the paired inferior chambers, known as the right and left ventricles, but we'll discuss their true anatomical placement. And the pump itself is the pump for the cardiovascular system, propelling our blood through a network of arteries, veins, and capillaries. And those network of blood vessels are referred to as these two different circuits. So again, with the two sides for each chambers, right and left in a pair for atria, right and left in a pair for ventricles, it's now the two right chambers, right atria and specifically the right ventricle, ultimately is propelling blood through the circuit of the lungs. Again, that's the job principally of the right ventricle. Whereas the left ventricle now has that job of propelling blood all around the body. So delivering that blood to the huge majority of our organs. Of course, the exception is pulmonary circuit unique to the lungs. So we see the placement of the heart within the, well, first of all, the thoracic cavity within the rib cage, specifically that medial section known as mediastinum, and the precise location, of course, the pericardial cavity. Comparing the position of the heart to those of the ribs, you can see spanning from about that third rib for the superior edge of the heart, which is known as the base. The base of any organ is always the broad end. So the superior base of the heart near the third rib, and it's here by the fifth rib, we have what's known as the apex. And again, the apex for any organ is the pointed edge of it. So that inferior apex, in this case, near the fifth rib. So going into the description of the placement of the heart, we have that approximate size, and again, that precise location within the pericardial cavity. And even though we name the chambers as right and left for atria, right and left for ventricles, what we call the right chambers actually make up the majority of the anterior surface. So the right chambers face predominantly forward towards the anterior side, making the left chambers pretty much face towards the posterior end. Again, the term apex is at inferior end, and you notice, of course, it's pointing down and to the left, of course, is at typical position. And the inferior apex is resting right on the diaphragm, connected right to that muscle. Again, we spelled it out with the previous picture, but the superior side, the base, that superior side near the third rib, and the inferior pointed apex, okay, lower down near the fifth rib. And we could just go back to the illustration to see that. So that superior broad base by rib 3, and the inferior angle to the left, there's the inferior point of the apex near the fifth rib. And just looking at the figure here, again, much of the anterior surface is composed of the right chambers, and we see very little of the left chambers from the anterior view. They make more of the posterior surface. So getting into the heart itself, there's three principal layers of tissue that form the structure of the heart. And again, you notice the prefixes here, endo, internal, epi, of course, at the surface, and myo must refer to the muscular portion. And again, the only type of muscle in the heart is that cardiac muscle. Only location of cardiac muscle in the body. So an alternate name for the epicardium, we've already learned previously, the visceral membrane bonded to the surface of the heart. And yeah, the visceral pericardium or the epicardium, synonymous terms. And then that second layer to the serous membrane lining the pericardial cavity is that parietal pericardium. So again, all together, it's truly these three layers here, and we can see it on the figure, that form the wall of the heart. So from the internal surface, lining the inside of the chambers, lining the hollow surface of the heart, the endocardium. The majority of the heart wall, again, comes from the layer of cardiac muscle tissue, so the myocardium. And the superficial layer of the heart, again, you notice here, epicardium, also known as visceral pericardium. Those three forming the wall of the heart, and all four chambers have those layers. There's variation, of course, in the thickness and makeup, but they all have the same three general layers, all four chambers. And then a detached separate layer, again, the parietal pericardium, the layer lining the internal surface of the pericardial cavity. So we previously looked at skeletal muscle, and there are a lot of similarities between skeletal muscle and cardiac muscle. So we want to point out these two very unique types of cell junctions, an intercalated disc and a gap junction. And both of these are absent in skeletal muscle. Skeletal muscle does not have either of these two types of cell junctions. Cardiac muscle has both, and they're both giving properties to make cardiac muscle very distinct, very unique. So the first one here, an intercalated disc, is truly just a very strong physical bond between neighboring cardiac muscle cells. So this allows for the cells to work as one large unit. All of the cells in a particular chamber are going to contract simultaneously because they have this physical linkage. One cardiac muscle cell will receive the stimulus to contract. It's bonded to its neighbors via these intercalated discs, thus pulling on the neighboring cell and stimulating the neighboring cells to contract. So this is one way to get the cells in a particular chamber to contract and work as a unit because they're bonded together via these intercalated discs. A second way to ensure that is via a gap junction. And a gap junction is another physical connection between cells. But what's unique is it also serves as an ion channel. So we have two neighboring cells. These two neighboring cells are connected together, but there is this open channel, and cardiac muscle cells could share now ions. And of course, specifically calcium's an important one, we know for muscle. Yeah, neighboring cardiac muscle cells could share calcium ions between them. So now we have this chemical and electrical linkage via the ions that could be shared between neighboring cells, and the stimulation could pass from one cell to that neighboring cell via the gap junction. So cardiac muscle has both of these two types of cellular junctions to ensure that the cardiac muscle cells in a particular chamber in the atrium or in the ventricle are working simultaneously, contracting to make a strong beat. So we could properly now propel the blood out of the chamber. And this slide here is really just kind of emphasizing more of the same. The thick layer of the heart is that middle myocardial layer. the cardiac muscle tissue. The endocardium and the epicardium are truly just lining on the inside or outside of that thick myocardium. And that's where you have the cardiac muscle tissue. And you see just an alternate name for it there. And again, cardiac muscle tissue has these two distinct type of cell junctions that are not seen in skeletal muscle. So cardiac has both of these. So if we go back to the idea of the circuits now, again, we can see it visually. The right ventricle, as we started up from the beginning, we described how the right ventricle's job is to pump blood to the pulmonary circuit, so pumping blood to the lungs. And the goal, of course, is the air that we breathe, the oxygen, could diffuse into the lungs, into the pulmonary circuit, and we're now enriching this blood with oxygen. And that's truly the purpose of the pulmonary circuit, is that we're going to add oxygen to the bloodstream. Whereas now the systemic circuit, this starts here now powered by the left ventricle, and we're now pumping that oxygen-rich blood all around our body to nourish the organs with oxygen. So on the systemic circuit, the purpose here is delivery of oxygen, but ultimately, at the end of the systemic circuit, that now deoxygenized blood is going to flow back into the right side of the heart, and then it's destined for another loop through the pulmonary circuit. So those erythrocytes that we discussed, of course, they could load up oxygen in the pulmonary circuit, and then they release the oxygen to tissues in the systemic circuit, and erythrocytes could just take that trip, of course, over and over through their lifespan. So again, we have 4 chambers in total to the heart. There's the pair for the atria, the pair for the ventricles. So we'll take a comparison between these two sets of chambers, and then we'll also start to see differences between the two different sides, the right chambers versus the left chambers. So starting with the atria, the term atrium means the entrance. So this is where blood flows from our veins. Various veins dump their blood into a particular atrium, depending on which side. The atria have a much thinner layer of cardiac muscle, a thinner myocardium, and this goes along with their purpose. The atria, their job is just simply to pump blood to the next chamber. And standing up with gravity, the atria have that just natural pull of gravity when we're standing up, so they really don't need to be that strong to propel blood just to the next chamber. Whereas now ventricles, the ventricles must be much thicker and stronger in their myocardium, because now, as we said, the ventricles are truly pumping blood through the arteries and either pumping from the right ventricle to the neighboring organ, to your lungs, which are just lateral to the heart, or especially the left ventricle, which has to pump blood from your left ventricle to your brain, to your fingers, your toes, all your muscles. So we have the thicker, stronger ventricles for the much larger job. So now in particular, taking a look at specifically the right atrium, because there's many adaptations for this single chamber. We have what's called the SA and AV nodes. These are known as the pacemakers, the natural pacemaker groups of the heart. And both of these are found in the wall of the right atrium. So embedded in the tissue of the right atrium. It's both the primary pacemaker and the secondary pacemaker. And that's what the SA node is the primary, and the AV node is the secondary. But both of those are embedded in the structure of the right atrium, and we'll discuss these soon. Blood from our systemic circuit, so blood from all around our body, with the exception of the lungs. So blood from your muscles, from your brain. Ultimately, all of that blood's going to flow through either three of these principal veins here. And all three of these major veins, superior vena cava, coronary sinus, and the largest vein in our body, inferior vena cava, all of these veins, which are all systemic veins, ultimately all three return their blood specifically to the right atrium. And one final trait about the right atrium is during our fetal development, there's actually an open hole, and you know this term here, foramen, a hole, the foramen ovale. This is an open hole from the right atrium, and it allows for blood to flow from the right atrium directly into the left atrium. But that's a trait, of course, during our fetal development. And shortly after we're born, as soon as we start to take our first breath, it'll hopefully start the healing process to close that foramen ovale. And what should be present by birth is known as a fossa ovalis, or shortly there after birth. In some cases, it does fail to close, and that can lead to complications. But the fossa ovalis is now the depression that's left after that foramen ovale closes up. So we can see that here again, the superior chamber with thinner walls, those are the atria. And in fact, in the right atrium, we can see that depression left of the fossa ovalis. We can see the large superior vena cava that's delivering blood from your head and arms, areas superior to the heart. We just see the cut remnant of the inferior vena cava. And from this view, we can't see that coronary sinus. It's on the posterior side, but there are three different veins that dump blood into the right atrium. We can see how the ventricles, especially the left ventricle, have a much thicker myocardium, much more muscle. And of course, taking a look at the right ventricle, which just has to pump blood to the lateral organ on each side, the location of the lungs. So the right ventricle has a smaller job to do, whereas now you can see that very thick, prominent myocardium surrounding the left ventricle. And this is the thickest, strongest myocardium of the heart. Because again, this left ventricle has to pump blood through the systemic circuit all around our body to deliver oxygen. The right ventricle is only pumping blood just to the lungs. So a quick look at the cytology of cardiac muscle. And you should notice here, Z discs and A bands and H zones. So again, a lot of similarities to skeletal muscle. And that's why we wanted to focus on the differences here. So intercalated discs and gap junctions. And again, this will look very familiar. The sarcolemma, the T tubules, the SR, the release of calcium. All right, so we'll move forward now. We have four chambers for the heart, and each chamber has its own valve. So at the exit point of each chamber, there's a valve, and the purpose of these valves in the heart, we'll discuss valves in the veins, valves in lymphatic vessels. Valves keep a fluid moving in one direction. So in this case, blood flows into the atria and blood flows and exits out of the ventricles. So the first set of valves, because again, keep in mind, we have the two atria and the two ventricles, right and left chambers. So a simple name is just between the atria and the ventricles. At the exit point of the atria, we have the right and left atrioventricular valves, and that's just spelled out down here with the alternate names. So between the right atrium and right ventricle It could be referred to as right atrioventricular. The right AV valve is composed of 3 different leaflets, meaning 3 different flaps of tissue. And there's the term tricuspid valve. It's another name for that. Whereas we see on the left side, the left atrioventricular valve. So now this makes sense. There's only 2 cusps of tissue that form the bicuspid valve, or even a very older term, but still acceptable, a mitral valve. And that's in place between the left atrium and the left ventricle. But in both cases, blood should only flow from the atria towards the ventricles. And if there is any backflow, that's an indication that there's a problem with these valves. The next set now, you notice between the ventricles and the arteries. So blood's forced out of the ventricles, passing through the arteries, starting to travel away from the heart. And to prevent backflow, that's now the importance of these semilunar valves. So we see they're named for the artery that they lead into. And in fact, with the newer editions of textbooks, it's not necessary to use the word semilunar. So just simply from the left ventricle, the aortic valve leads to the aorta. From the right ventricle, the pulmonary valve leads into that pulmonary trunk. So here's a view of the heart, you notice, with the superior atria removed. So we're able to look right down onto the atrioventricular valves, as well as we see the placement of the semilunar valves. And in particular now with the atrioventricular valves only attached to the AV valves, we have these tendon-like structures, and hence the name, chordae tendineae. And in turn, we have the fold of the atrioventricular valve, whether it's bicuspid or tricuspid, they both have this. Inferior to the leaflets of the valve, we have the tendon structure, the chordae tendineae. And in turn, the chordae tendineae connects to what's called papillary muscle. And this is a particular feature of muscle located in the inferior ventricles. So from this view, the atria would be up top, the ventricle, of course, inferior. And when the muscle in the ventricle is relaxed, the tension on the chordae tendineae is relaxed, and the pressure and force of blood can and does travel down through the open AV valves into the ventricles. However, when the ventricles are now contracted, the papillary muscles contracted, putting tension on the chordae tendineae. And just like the illustration shows, blood is forced against the AV valve, but they're prevented from inverting, and blood cannot flow up into the atria. Instead, that blood and the pressure is forced against the semilunar valves, and blood could flow out of the heart, out of the ventricle. So again, the chordae tendineae, that's the unique feature of the atrioventricular valves. Not at all or never the case for the semilunar valves. And in fact, the sounds of the sets of valves closing, that's the major sound that's oscillated, that's listened to by a physician when they're moving a stethoscope around your chest. But that's not an important detail for us. It's just more to mention. As well as the placement. So the next topic we want to move into now is what's known as the cardiac cycle. And it's really characterized by these 2 principal phases. So every chamber in the heart, all 4 chambers, both atria, both ventricles, there's a period of systole, And this is the period where the cardiac muscle tissue is contracting, reducing the volume of the chamber, increasing the pressure within. And systole is the force to propel the blood out of the chamber. And the second phase now, diastole, the muscles allowed to relax. As the chamber expands and the pressure decreases, this is now the period of time in which the chamber fills with blood. So of course, cycling through these two phases, filling and pumping, this is the cardiac cycle. However, the four chambers are never, or I should say, not exactly in sync. So we'll see that there's a period of time when the atria are in systole, and there's a period of time when the ventricles are in systole. So we have this diagram here, and without getting into the physiology too much, we see right at the start of the cycle, the pink color is indicating systole contraction. And yes, the atria begin the cycle with a period of systole, a period of contraction. This is now forcing blood through those open atrioventricular valves to fill the ventricles. And it's not until a delay in time until the ventricles begin their phase of systole. So the ventricles now start to contract slightly later after the atria already have. So that's our main takeaway, because we don't want to get into the physiology. We're going to look at the underlying anatomy, of course. But at the start of the cycle, the atria contract in systole. There's a delay in time before the ventricles begin their phase of systole. And then ultimately, of course, one of your physio instructors will fill you in on the rest of this. And we'll move on to continue the cycle. But as I mentioned, instead, we're going to look at the underlying anatomy that really dictates this physiology. So just take a moment to take a look here at the function of the heart, because we have the paired chambers, right and left atria, right and left ventricles, and the two sides are working simultaneously. The two atria contract, the two atrioventricular valves open and close in rhythm, the ventricles contract and relax in rhythm. So we have the function after we're born, and that's key to note. This is the purpose and function after we're born. Fetal circulation is very distinct and very unique. But we notice flowing into the right side of the heart, we have this deoxygenated blood. The oxygen has been consumed by our tissues and organs, so the oxygen concentration is much, much lower. And that blood's flowing into the right side of the heart, from the right ventricle specifically, flowing on to the lungs. And there, the erythrocytes can then reload with oxygen. And now, fresh from the lungs, rich in oxygen, now that blood flows into the left chambers of the heart. And this oxygen-rich blood flowing through the left chambers out to the aorta. And now the aorta, our largest artery, is going to deliver and divert blood to multiple small arteries and deliver that oxygen-rich blood to our various tissues and organs. But again, the right and left sides are functioning simultaneously. But the right and left sides have a very different purpose. So getting near the end of this chapter and finishing up this section, we're now going to talk about the structures that's involved in really dictating that cardiac cycle. And this involves various pacemakers and what's known as conducting fibers. So together, the pacemakers and the conducting fibers make up this intrinsic conduction system. And intrinsic means from within the organ. So there are structures, pacemakers and conducting fibers. These are both within the tissue of the heart, within the myocardium, and the stimulus to contract comes from these pacemakers that are within the myocardium. So a heart, a living heart, if vivisected from an organism, will continue to beat for a short period of time. If it's removed from the live specimen, the heart stimulates its own contraction. Think about that intro screen that we had for this lecture. So the pacemakers, these specifically are the cells that have the ability to produce an action potential to produce an electrical current. Depending on the speed of the pacemakers are the fastest nodal cells. So we have the term AV node, SA node, and within each node, the fastest nodal cells get the distinction of being a pacemaker. Thus, the fastest nodal cells, the pacemaker cells, will establish the basic rate of the heart's contraction. The pacemaker cells are the ones that are producing the electrical current, whereas conducting fibers, their job is not only just to spread that electrical current from the pacemakers throughout all of the cardiac muscle in all four chambers. So really distributing that function that started at the pacemakers and just distributing that throughout all four chambers myocardium. So we have the two categories here, primary and secondary, the two different types of nodal cells. And again, they have this unique ability of producing an electrical current, and that's the term depolarization. So producing this positive charge. And this positive charge, this action potential, generated by the primary pacemaker, the SA node. And it gets the distinction as primary. It works at a faster rate when compared to the slower atrioventricular node. So in a healthy heart, the primary pacemaker sets the basic rate of contraction, the SA node, and the AV node does serve as a backup. You can imagine if there's a heart attack, if there's injury and death to the primary pacemaker, the secondary pacemaker will stimulate the ventricles to contract, and that heart, of course, will be greatly compromised, but it will be able to circulate blood at a reduced rate. So we have a description in the text here, but let's just skip to the figure. So putting this together, this intrinsic conduction system, again, we see both of the nodal groups, sinoatrial and atrioventricular. Both are embedded in the structure of the right atrium. So it's these two areas here that are the pacemakers. Again, the SA node is the primary pacemaker, and we see that there's a direct interaction via these conducting fibers known as internodal pathways. So the SA node does signal and stimulate and control the AV node in a healthy heart. And it's now from the AV node, there's a series of many more conducting fibers, the bundle of His, branching to the right and left bundle branches. And then ultimately these Purkinje fibers, the Purkinje fibers in turn would stimulate the cardiac muscle of the ventricles. So the myocardium of the ventricles is stimulated by these Purkinje fibers. So if we now relate this back to our cardiac cycle, and you could go back a few slides, of course, to see that view. At the beginning of the cardiac cycle, the atria contract, And this makes sense due to this anatomical structure because the atria, specifically the right atrium, contains the pacemaker. So the atria will be in systole, their contraction phase, right at the beginning of the cardiac cycle. And the reason again is because the atria contain the pacemakers, and that very brief delay in time it takes for this electrical signal to travel all the way through to the Purkinje fibers to reach the ventricles. There's that delay, and then ultimately the ventricles start their phase of systole slightly thereafter. The atria have already relaxed. So again, it's this intrinsic conduction system, this anatomy, is what, of course, dictates the physiology seen here in the cardiac cycle. And the figure shows that really well. And here it was just being described in the text. So the SA node able to signal to the AV node. So we have that connection of the internodal pathway. SA nodes able to signal to the secondary pacemaker. Of course, between cardiac muscle cells, again, cardiac muscle cells contain both the gap junctions, the ion channels between them, and the strong physical connections of intercalated discs. So this way, neighboring cardiac muscle cells are able to stimulate one another. And then, of course, from the AV node, again, following the steps to reach the Purkinje fibers, and then ultimately Purkinje fibers stimulating myocardium of the two ventricles. And the full image shows this here. So with the primary pacemaker embedded in the superior portion of the right atrium, and this purple tint is now illustrating the spread of the depolarization, the spread of that positive charge, leading to the contraction of both atria. And again, that brief delay in time for signal to travel down to the ventricles via the Purkinje fibers, and then ultimately the stimulation of the ventricular myocardium. So again, it's this anatomical arrangement here is what dictates the timing and the physiology of the cardiac cycle. So the heart contains its own intrinsic conduction system, and a isolated living heart can continue to beat for a short period of time until the organ does expire. So the recording of the electrical voltage that spreads from the pacemakers from the conducting fibers can be recorded even at the level of our skin. And that's what makes a heart monitor, or you see the term here, electrocardiogram. EKG was the original name for it. An electrocardiogram is now just a measurement of only two things. There's time going by. So time is going by. And here on our y-axis is truly just a measurement of the voltage. Spell that right. Measurement of the voltage. So those are the only two things that are on this graph, just time and voltage. But based on this graph and an understanding of the graph, we could interpret and infer and make a diagnosis about the health of the heart. So just a quick look here. We have this graph of the ECG, And there's just the different segments. So what's known as a P wave, the QRS wave complex, the T wave, the various segments, and again, more of a topic for physiology, the measurements and the recordings. And just as an example, of course, knowing and understanding the graphs could help us interpret that this now is showing a accelerated fast heart rate. The appearance of a heart attack has its typical rhythm. Or in here, you notice, for example, this is showing an issue at the area of that AV node. So it's known as a heart block. So again, interpreting the signal, you can infer so much about the function of the heart. And again, just FYI. So yes, the heart does have its own intrinsic conduction system. However, it is now modified, that's the key word, so it could be regulated, upregulated or downregulated by what's known as the autonomic nervous system. So our nervous system, our brain, of course, has now a way to speed up the heart rate or slow down that intrinsic system. And the autonomic nervous system has two general divisions. So it's known as parasympathetic and sympathetic, commonly referred to as sympathetic, fight or flight, the parasympathetic, the rest and digest. And it influences each of the pacemakers by, of course, producing these neurotransmitters. So ACH, acetylcholine, norepinephrine, just using the abbreviation there, NE. With our parasympathetic rest and digest system, our heart's put into a more relaxed state. So the function of the pacemakers and thus the heart is decreased. So to allow for a slower resting heart rate and reducing the strength of contraction. Whereas the sympathetic, the fight or flight, when we need more blood pumping, where we're using more energy, consuming more energy, using our muscles. Norepinephrine, or just simply NE for our purposes, will lead to that increase in the heart rate to really get the blood pumping and have the heart pump with more force. So per beat, we're propelling more blood. All right, so that'll finish up this second part of the system, the pump, the heart structure.